You know those amazing pictures of corn? Maize, right? Where the kernels are all striped and spotted with these really vibrant colors?
Oh yeah, the maize kernels. Classic example.
Well, it turns out those patterns aren't just, simple genetics like we learned in intro bio. They're actually visual proof of genes literally jumping around inside the corn's DNA.
That's exactly it. We're diving into transposable elements today, or transposons as they're often called. That early work on maize, mostly by Barbara McClintock, really blew the lid off how dynamic genomes actually are.
It wasn't just neat rows of genes staying put.
Not at all. So our goal here is to kind of walk through the key ideas from this chapter, look at the different types of these mobile bits of DNA, figure out how they move, the mechanisms involved, and really grasp their huge impact on genetics and evolution. And they're everywhere: bacteria, plants, us.
And when you say everywhere, the numbers are just mind-blowing. Like in maize, something like 85% of its DNA comes from these things.
85%. It's incredible.
But here's the real kicker for you listening. Almost half of your DNA, the human genome, 44%, it's made of these jumping genes or their fossil remnants.
Yeah, it really drives home that genomes aren't static. They change, they evolve, and these elements are major players. So to get a handle on it, we need to break down how they move. There are basically three main ways, three categories of transposition.
Okay, let's start with category 1, cut and paste. Sounds pretty straightforward.
It is. Conceptually, the transposon literally gets cut out of its spot in the DNA.
Snip.
Right, snipped out by an enzyme called transposase, which usually the element itself carries the gene for. Then it just gets pasted into a new location in the genome. Simple excision and insertion.
And we find these kinds of transposons.
Where?
Oh, all over. In bacteria, those little IS elements are a prime example. And in eukaryotes, too, like the ACD system and that maize we were just talking about.
Okay, cut and paste. Got it. What's mechanism number 2?
Number 2 is replicative transposition. Think copy-paste, not cut-paste.
Ah, okay, so the original stays put. Exactly. The original element doesn't move, but it gets replicated, and that new copy is inserted somewhere else. So the net result is you go from one copy to 2 copies in the genome.
And you mentioned this one's only in prokaryotes.
That's right. Historically, that's where we see it. The TN3 element in bacteria is the textbook example for this one.
Interesting. Okay, and the third way they move, retrotransposition. This sounds different. Involves RNA, right?
Yeah, this is where things get a bit wild. It's kind of a reverse flow of genetic information. You see this only in eukaryotes.
So what happens?
Well, the transposon's DNA sequence is first transcribed into an RNA molecule, like making a message.
Standard transcription.
Right. But then an enzyme called reverse transcriptase uses that RNA message as a template to make a DNA copy.
Going backwards, RNA to DNA, hence retro.
Precisely. And then that new DNA copy gets inserted back into the genome somewhere else. This is the mechanism for retrotransposons and retroposons. It's all about that RNA intermediate.
Wow. So 3 distinct ways. Cut and paste, copy paste, and this RNA intermediate retro way. But do they leave any kind of signature? Like, can we tell where they've been?
Yeah, there are clues. Structurally, many transposons have what are called terminal inverted repeats, short DNA sequences at their ends that are, well, inverted copies of each other. The transposase enzyme often recognizes these.
Okay.
But the real calling card, the thing you almost always see after an insertion, is a target site duplication.
Target site duplication. What's that?
So when the transposon inserts, the enzymes involved make a staggered cut in the host DNA at the target site. The transposon slots in, and then the cell's repair machinery fills in the gaps on either side. Because the initial cut was staggered, filling the gaps creates short identical sequences flanking the newly inserted transposon. It's a direct repeat of the DNA sequence that was originally at the insertion site.
Ah, I see, like a little footprint left behind.
Exactly, a molecular footprint.
Okay, let's zoom in on bacteria for a bit. You mentioned IS elements, insertion sequences.
Right. These are the simplest bacterial transposons. Really minimal. They're small, and typically they only code for the transposase enzyme they need to move themselves. That's it.
How are they first found?
Through mutations, actually. Scientists noticed some mutations in E. coli weren't stable. A gene would suddenly stop working.
Because an IS element jumped into it?
Exactly. But then, sometimes, the gene would spontaneously start working again. The IS element had jumped back out, restoring the original sequence. It showed this mobility directly. It's the perfect bacterial cut-and-paste example.
But their impact is bigger than just causing reversible mutations, isn't it? You mentioned recombination.
Oh, definitely. They play a huge role. If you have identical IS elements located on, say, a plasmid and also on the main bacterial chromosome, they provide regions of homology. The cell's recombination machinery can then act on these matching sequences.
And swap bits of DNA or join things together.
Precisely. This is how the F plasmid, the fertility plasmid, can integrate into the E. coli chromosome to create those HFR strains, high frequency of recombination strains. It's IS elements mediating that fusion. It's also key for combining different genes, like antibiotic resistance genes, onto one plasmid.
Which leads us to composite transposons, right? Like TN5.
Yes, exactly. Composite transposons are fascinating. Imagine two IS elements happen to insert near each other in the bacterial DNA.
Yeah.
Sometimes they can then start acting as a single unit. The transposases from the outer IS elements recognize the outermost ends, and they move not just themselves, but the entire chunk of bacterial DNA located between them.
So they pick up cargo.
They pick up cargo DNA. And this is hugely important medically because that cargo often includes genes conferring resistance to antibiotics, things like kanamycin resistance, CANAR, or streptomycin resistance, Structer. TN5 is a classic example carrying CANAR.
And that's how resistance spreads so fast. These composites jump onto plasmids.
That's a major route, yes. They can easily hop onto plasmids, which then spread rapidly through bacterial populations. Sometimes the flanking IS elements aren't even identical. Like in TN5, one is functional, IS50R, and the other IS50L has mutations, but they still work together.
Wow. Okay, that's a real problem for medicine.
Yeah.
Now, before we leave bacteria, you mentioned TN3 and the replicative mechanism. How does that work? It sounds more complex.
It is a bit more involved. TN3 is a replicative transposon, and it carries genes not just for transposase, but also for another enzyme called resolvase, and usually a resistance gene like beta-lactamase for ampicillin resistance.
Okay, transposase, resolvase, resistance. What's the process?
It happens in two main stages. Stage 1. The transposase acts. It doesn't cut the TN3 out. Instead, it helps fuse the DNA molecule carrying the TN3, like a plasmid, with the target DNA molecule, another plasmid maybe.
They merge.
They merge into one larger circle called a cointegrate. And crucially, during this fusion process, the TN3 element itself is replicated. So the cointegrate ends up having two copies of TN3, one at each junction between the original donor and target DNA.
Ah, so that's the replicative part. You now have two copies where you started with one.
Yeah.
But they're stuck together in this cointegrate.
Exactly. That's where stage 2 comes in, involving the resolvase enzyme, which is encoded by the TNPR gene on TN3. The resolvase recognizes a specific site within each TN3 copy called the res site.
Okay.
And it performs a site-specific recombination event between the two res sites on the cointegrate. This neatly resolves the cointegrate back into the two original plasmids, but now each plasmid carries its own copy of TN3.
Clever. So fusion and replication, then resolution.
Right. And there's another neat trick. The resolvase protein also acts as a repressor. It dials down the transcription of both the transposase and its own gene.
Why do that?
It keeps the TN3 element relatively stable most of the time. It prevents it from constantly transposing, which could potentially harm the host cell. It's a form of self-regulation. Spread, but don't go crazy.
Right. Makes sense. Okay, let's shift gears to eukaryotes now.
Yeah.
Back to maize and that cut and paste system, the ACDs system.
Yes, Barbara McClintock's work. It's a beautiful illustration of autonomous versus non-autonomous elements.
Explain that. Autonomous versus non-autonomous.
Sure. EASE, which stands for Activator, is the autonomous element. It's complete. It contains the gene for a functional transposase, so it can direct its own transposition, it can cut itself out and paste itself somewhere else.
It's independent.
Right. Now, Ds, for dissociation, is non-autonomous. Think of it as a broken AC element. It typically has deletions within the transposase gene, so it can't make its own enzyme.
So it's stuck.
It's stuck, unless there's a functional AC element somewhere else in the same cell nucleus. If AC is present, it provides the transposase enzyme, which can then recognize the ends of the Ds element and move it.
Ah, so Ds relies on AC for the machinery. It's like borrowing the tools.
Exactly. Ds has the cut here signals, but not the scissors. AC brings the scissors.
And how does this relate to the spots on the corn kernels? You mentioned breakage.
Yes, the Ds element got its name dissociation because its movement can actually cause chromosome breaks. Let's take the C locus, which controls kernel color. Imagine a kernel should be colored, but there's a dominant inhibitor gene, let's call it CI, located further out on the same chromosome arm.
Okay, so CI prevents color.
Right. Now, if a Ds element inserts near the C locus, and then an AC element tells it to move, Sometimes when Ds tries to excise, especially during chromosome replication, it causes a break at that location.
A physical break in the chromosome.
Yes, and if the break happens between the centromere and the CI gene, the end part of the chromosome arm, the part carrying CI, gets lost because it doesn't have a centromere to be pulled along during cell division.
It just floats away.
It gets lost from the cell lineage. So in that cell and all its descendants, the CI inhibitor is gone. Without the inhibitor, the C gene can now function, and those cells produce pigment.
Creating a patch of color on an otherwise colorless kernel.
Exactly. That's the mosaicism. Each spot or stripe represents a lineage of cells that lost the inhibitor gene due to a Dis-mediated chromosome break, triggered by AC. It's a direct visual readout of transposition.
That's incredible. Okay, another eukaryotic example. P elements in fruit flies, Drosophila, associated with something called hybrid dysgenesis.
Right. P elements are another cut-and-paste system. They were discovered because of this strange phenomenon, hybrid dysgenesis. Basically, when you did specific crosses between different lab stocks of flies, the offspring would have all sorts of problems. High mutation rates, chromosome abnormalities, often sterility. It was like the genome was falling apart.
But only in specific crosses.
Yes, that was the key clue. It only happened reliably when males from a strain with P elements, P strain, were crossed to females from a strain that historically lacked P elements, M strain.
P males, X, M females equals problems. What about the reverse cross, M males, X, P females?
That cross was usually fine. The offspring were normal. This asymmetry pointed toward something weird happening with regulation.
So what's the regulation? Why does the direction of the cross matter?
It boils down to two layers of control. First, P element transposition is normally restricted to the germline, the cells that make sperm and eggs. It doesn't happen in the body's somatic cells.
Why not?
It seems to be due to RNA splicing. The P element gene makes a pre-mRNA, and only germline cells can splice it correctly to produce the active transposase enzyme. Somatic cells splice it differently, making a non-functional protein.
Okay, so that protects the fly's body. But what about the hybrid dysgenesis in the germline of the offspring from that P male XM female cross?
That's the second layer. Cytoplasmic control. Females from P strains, the ones that have lived with P elements for generations, package specific small RNA molecules called pRNAs into their eggs.
pRNAs, Piwi-interacting RNAs.
Correct. These pRNAs are complementary to P element sequences, and they act like a silencing mechanism, basically shutting down P element activity in the developing embryo.
Ah, so in the P male XM female cross, the sperm brings in the P elements.
Right.
But the egg from the M female doesn't have those protective pRNAs because her strain never had P elements to silence.
Exactly. So the P elements inherited from the father find themselves in a permissive environment. The egg cytoplasm lacks their repressors. They go wild in the germline of the offspring, causing all those mutations and chromosome breaks. That's hybrid dysgenesis.
But in the reverse cross, M male X P female, the P female's egg does have the pRNAs. So even if the offspring inherit P elements later, they're kept in check from the start.
Precisely. It's a beautiful example of cytoplasmic inheritance and genome defense mediated by small RNAs. Very tightly controlled.
Wow. Okay, that's sophisticated control. Now let's move to that third mechanism, retrotransposition. You said it relates to retroviruses.
Yes, understanding retroviruses was key. Retroviruses, like HIV for instance, have an RNA genome. When they infect a cell, they use their own enzyme, reverse transcriptase, to make a DNA copy of their RNA.
The reverse flow again.
Right. Then another viral enzyme, integrase, inserts that DNA copy into the host cell's chromosome. That integrated DNA copy is called a provirus. Retroviruses typically have three main genes: GAG for structural proteins, POL for reverse transcriptase and integrase, and NV for the envelope protein that lets them infect new cells.
Okay, so how does that relate to transpose?
Well, the first class of retrotransposons discovered look remarkably like integrated retroviruses. They're called retrovirus-like elements, or LTR retrotransposons.
LTR.
Long terminal repeats. These elements have long direct repeats at each end, similar to proviruses. They also contain genes related to GAG and POL, including reverse transcriptase and integrase. Examples are the Taiwan element in yeast or the copia element in Drosophila.
Well, what's the difference from a real retrovirus then?
The crucial difference is they lack the N gene. Without the envelope protein, they can't make infectious viral particles to escape the cell and infect others. They can only copy themselves and jump to new locations within the same cell's genome, or like trapped viruses.
Genetic ghosts of viruses stuck inside.
Okay.
What's the other type of retrotransposon?
The other major class are the retroposons, also called non-LTR retrotransposons. These are different structurally. They lack the long terminal repeats.
No LTRs. What do they have instead?
Their signature feature is often an AT-rich sequence at one end. This comes from the poly-A tail that gets added to most eukaryotic messenger RNAs. It's a telltale sign that they moved via an RNA intermediate that was processed like a regular cellular mRNA. The main types you hear about are lines and signs.
Lines and signs. We'll get to those in humans soon. But is there anything particularly interesting these retroelements do besides just copying themselves?
Sometimes, yeah. In Drosophila, for example, the cell has actually co-opted a couple of specific retroposons called HETA and TART.
Co-opted them?
Yeah.
For what?
For maintaining the ends of chromosomes, the telomeres. You know how DNA replication tends to shorten chromosome ends?
Right, the end replication problem.
Well, most eukaryotes use an enzyme called telomerase to counteract that, but Drosophila doesn't seem to use telomerase in the same way. Instead, these HETA and PART retroposons specifically transpose onto the chromosome ends, adding DNA sequences back and essentially healing the telomeres.
No way. So they turned these jumping genes into essential maintenance workers.
Pretty much. They went from potentially being genomic parasites to being indispensable parts of the fly's chromosome biology. Evolution is clever.
That is really cool. Okay, so let's bring it home to humans. That 44% statistic is still buzzing in my head. Are these things still active in us?
Most of that 44% represents ancient insertions that are now inactive, mutated, broken, essentially genetic fossils. But yes, some are still active, particularly one type of line element.
Lines, long interspersed nuclear elements. Which one?
The main active one is called L1. It's the most abundant transposon in our genome by mass, even if not by copy number. And importantly, it's autonomous.
Meaning it can move itself.
Yes, L1 elements are about 6 kilobases long, and they encode two proteins, ORF1 and ORF2. ORF2 has both a reverse transcriptase activity and an endonuclease activity, an enzyme that can nick DNA.
So it have the whole toolkit. How does it move?
Briefly, the L1 element's DNA is transcribed into RNA. That RNA is translated into the ORF1 and ORF2 proteins, which seem to stay physically associated with the RNA molecule they came from.
Okay, the team sticks together.
Yeah. Then, this complex finds its way back into the nucleus. The ORF2 endonuclease nicks the chromosomal DNA at a target site. Then, using that nicked DNA as a primer and the L1 RNA as a template, the ORF2 reverse transcriptase synthesizes a new DNA copy of the L1 element directly into the chromosome.
Wow, target primed reverse transcription.
Exactly. The result is a new L1 insertion, often with a target site duplication like we discussed earlier. Now, these insertions are relatively rare on a per-generation basis.
But they happen.
They do happen. And when an L1 inserts into or near an important gene, it can disrupt it and cause genetic disease. There are known cases where L1 insertions have caused hemophilia, for instance. They are a source of ongoing spontaneous mutation in humans.
Okay, so L1s are the active autonomous players. What about SINE-LS? Short interspersed nuclear elements.
Right. The most famous SIN is the ALU element. These are much shorter than L1s, only a few hundred base pairs. But they are incredibly numerous. There are over 1 million ALU copies scattered throughout your genome. They make up about 11% of our DNA.
1 million copies. Are they active?
They are transpositionally active, yes. ALU elements do occasionally insert into new locations and can cause disease. But here's the catch. ALU elements are non-autonomous.
Ah, like the D's elements in maize. They can't move themselves.
Correct. ALU elements do not encode any proteins, certainly not a reverse transcriptase or endonuclease. They are entirely dependent on borrowing the machinery made by other elements.
And which elements do they borrow from?
Primarily the L1 elements. ALU sequences are thought to be mobilized by the ORF2 protein provided by active L1s.
So ALU elements are basically parasites on the L1 elements, which are themselves sort of genomic parasites.
That's a great way to put it. It's layers of genomic parasitism. ALU is incredibly successful, spreading to over 1 million copies by effectively hijacking the L1 machinery. It's a fascinating evolutionary dynamic happening within our own DNA.
Incredible. Okay, stepping back then, what's the big picture significance of all these transposons across life?
Well, several key things. First, as we've seen, they are inherent mutagens. By inserting into genes or near genes or by causing breaks during excision, they constantly generate new genetic variation for better or worse. Think about mutations in the white gene in Drosophila. Many were caused by transposon insertions.
Oh, mutation source. What else?
Second, they've become invaluable tools for geneticists. Once we understood how they work, especially elements like P elements, scientists figured out how to hijack them. We can engineer transposons to carry specific genes we want to insert into an organism's genome.
Like gene therapy vectors. Or for research.
Yeah.
Exactly. P elements were crucial for early Drosophila transformation, carrying genes like Rosie. Other systems developed later, like piggyback or sleeping beauty, are used widely in various organisms, including potential therapeutic applications in mammals, as vectors for gene delivery.
Turning the tables on the jumping genes.
Yeah.
Nice. And third,
Third, they are major architects of genome structure. They don't just cause small mutations.
Yeah.
Think about having multiple copies of the same transposon scattered around the genome. If the chromosomes misalign during meiosis, perhaps pairing up between two transposon copies on different parts of a chromosome or on sister chromatids.
you could get unequal crossing over.
Precisely. If two transposons are oriented in the same direction, unequal crossing over between them can lead to the deletion or duplication of the entire segment of DNA located between those transposons. These can be huge chunks of the chromosome. So transposons facilitate major chromosome rearrangements, which are a powerful force in evolution.
Wow. Deletions, duplications, just by having these repetitive elements lying around. Right. They provide the substrate for these large-scale changes.
Okay, so quite a journey. We started with colorful corn kernels.
Yeah, those McClintock observations.
And ended up seeing how these mobile DNA sequences use basically three core strategies: cut and paste, replicative copy-paste, and the RNA-based retrotransposition to move around. We saw key discoveries in bacteria, maize, flies.
And how that led us to understand the ongoing activity of lines and signs, these ancient travelers, still shaping our own human genome today.
It really paints a picture of the genome as this incredibly dynamic, fluid entity, not just a static blueprint.
Absolutely. It's constantly being reshaped, and transposons are a major part of that story.
So here's something for you, our listeners, to think about. We mentioned that the actual rate of new mutations caused by transposons seems quite different between species, right? Like it's thought to be relatively high in flies, but much lower, much more tightly regulated in humans.
Yeah, our L1s and Alicus move, but maybe not as frequently or as disruptively on average as P elements could in those dysgenic crosses. We have defenses, like those RNAs and maybe other mechanisms too. Most of our 44% really are just quiet fossils.
Right, so here's the question. Why is it that these elements in our genome, which are mostly inactive and heavily regulated, can still, when they do occasionally move or get reactivated, be such potent sources of genetic novelty and major genome rearrangements? Compared to, say, systems in other organisms where transposition might be more frequent, but perhaps less impactful per event. Is there something about the history or the regulation itself that makes our sleeping giants capable of such significant changes when they occasionally wake up? Something to mull over.